Dr. Lazarov, thanks so much for your time today. It's been great visiting with you today at the University of Illinois in Chicago. You're engaged in some fascinating research, and I wonder if you'd just briefly explain to us the kind of projects that you're interested in and the main focus of your research here.
Of course. Thanks for visiting us. It's been a pleasure. We're interested in understanding what goes wrong in the brains during embryogenesis in the brains of Down syndrome individuals. We're looking mainly to understand the deficits in neuronal development and extrapolate how we can actually fix those deficits in the brain during development. We're using different models of stem cells that we've been generating of Down syndrome patients in order to understand how neurons develop and how they populate the brain of those individuals.
Fascinating. So in examining these induced pluripotent stem cell structures, can you correlate their development with embryonic development on a week-to-week sort of basis, or how does that happen?
Right. So the idea is to use cells that are derived from patients and then use them as neurons, as brain cells, and try to understand what goes wrong in the development of those neurons. So certainly, the ability to follow up in a timely manner and understand their development, or where in neuronal development we see deficits, that would tell us what the deficits are and how we can correct them.
Great. So in identifying where the deficits are, the next step then would be to try to discover potential therapies that would reverse that.
That's right. That's absolutely the ultimate goal, is to try to identify the deficits and then try to correct them.
And are you thinking that might be possible to do during the developmental period, during the fetal development, or after birth? Or
that's our hope, that we can actually correct those deficits by the time that the child is born. The deficits are corrected. That's the goal.
Great. And if these deficits are occurring during the fetal period and there's no intervention possible at that point, is it possible to rescue that development after birth, or is an individual fairly well locked into the neurological structures that they have at birth at that point?
Well, ideally, yes. The hope is that if we understand what the deficit is, we're able to rescue it even postnatally. Or at least compensate for the deficit. Yes, certainly. So potentially that would be possible. Depends on the deficit, but potentially that would be possible.
Yeah, and so if you're able to rescue that development during the prenatal period, there's still, in the case of Down syndrome or trisomy 21, the presence of the extra chromosome throughout the person's life with the same sort of metabolic imbalance. I would presume then that therapy would need to continue, and it's probably impossible to say for sure because there's so many things we don't know at this point, but those therapies would probably need to continue then throughout the lifetime? Or?
My assumption is yes. It will probably take a combination of therapies to overcome all the deficits that result from the three chromosomes. That said, our main focus is cognitive impairment. So once our main target is the mental retardation. Once we fix the cognitive deficits, then we can take care of other abnormalities that affect those individuals. Yes.
All right. Well, your research really is fascinating and very innovative. And thank you so much for everything that you're doing on behalf of all those who have children with Down syndrome and will have in the future. I very much appreciate your efforts, and we look very much forward to staying in touch and seeing what results you produce in the future.
Thank you, and I would like to thank the Foundation for supporting our research. It's been a great contribution, very important one, and it's thanks to your foundation that this research is possible. So thank you too.
It's our pleasure, and thank you so much again.